Hello, dear students. Today I am going to discuss about the second part of the history of microbiology. In the first video, I have already discussed about some of the renowned contributions by the scientists who laid the foundation of the microbiology. So their contributions opened the new inventions, gates for the new inventions and discoveries, and also inspired some of the scientists to work on this field, and their contribution also was greatly, most of the achievements made by them cannot be ignored. So today I'm going to make the second part of the history of microbiology. After the work of the Robert Koch and Louis Pasteur, they attracted so many scientists and their disciples who worked on this field, and also led to the discovery of some of the new bacteria. First important scientist which I would like to mention is about the Gerhard Henrik Armeur Hansen, whose life period was from 1841 to 1912. In 1874, Gerhard Henrik Armeur Hansen, a Norwegian scientist, discovered Mycobacterium leprae as the causative organism for leprosy, defying the hereditary affliction of the disease. Defying the hereditary affliction theory of the disease. Earlier, it was believed that the leprosy disease is caused by some of the hereditary abnormalities and is transferred from one person to another person. So he was the first scientist who gave that basically the leprosy is caused by the bacterium called as the Mycobacterium leprae. Next scientist was Albert Ludwig Sigismund Neisser, year 1855 to 1916, who was a German physician. Albert Ludwig Sigismund Neisser, in 1879, he described the gonococcus. The history of sexually transmitted disease is thought to date back to earliest times, and many ancient texts describe conditions that may be those of syphilis and gonorrhea, which at one time were thought to be the same disease. So basically the difference between the sexually transmitted disease mostly caused by two important pathogen, that is the Neisseria gonorrhea and syphilis, that is Treponema pallidum, are the two separate agents. But previously, it was seen that it was very difficult to distinguish that which organism is responsible. So, Neisser first described the bacteria Neisseria gonorrhoeae, and then the the department was able to differentiate between the two important pathogen, and also it helped in the treatment because the treatment of syphilis and gonorrhea need different regimen. Next, one important story about the credit for the discovery of leprosy bacillus was between the Neisser and Neisser and Hanser, and the basic difference was that the Gerhard Hansen initially discovered the rod-shaped bodies in leprous nodules in 1873, but could not stain them, while Albert Neisser, using staining techniques, successfully stained the organism in 1879 and published his findings, claiming priority. Hansen had already done significant work and was able to stain the bacilli with the help from others, leading to priority dispute that continued for years. So during the same time period, both Hansen and Neisser were working on the leprosy bacillus. Gerhard Anderson was the first scientist who discovered the rod-shaped bodies in 1873, whereas Albert Neisser gets the credit of staining them in 1879 and also published his findings, taking the credit of finding out the Mycobacterium leprae microorganism. Now, what happened that in 1873, where Gerhard Hansen described the leprous bacillus, Albert Neisser visited Hansen and obtained tissue samples and used improved staining techniques to stain the organism. He published his work claiming he was the discoverer. The controversy and credit. Back and forth publications. Hansen responded to Neisser's claim in 1880 with his own paper detailing his research. Neisser in 1881 argued that his work was more important because he was the one to connect the bacteria to leprosy, even though he admitted he wasn't the first to see them. So here the dispute was basically regarding the credit for discovery of the leprae bacillus. The Hansen was the first to isolate the bacillus, and therefore in Neisser also admitted that he was not the first to isolate, but he stained it and he was able to stain the bacteria. So it there was a long-standing dispute regarding the who was the discoverer of the bacillus. So controversy went on the years, and Neisser remained resentful that he had not received more recognition. Modern concession is that today Hansen is generally acknowledged as the true pioneer for his initial discovery and his work in demonstrating the infectious nature of the disease, while Neisser is credited for providing the first good description of the bacilli using his superior laboratory techniques. So, still the leprae bacillus is also called as Hansen bacillus also in respect of Hansen. One of the important unethical aspect that was there that Hansen was upset by Neisser's action because he published his findings in the year 1879 about the staining techniques. While when he visited the lab of Hansen, he was greeted very warmly by Hansen. Neisser was greeted very warmly by Hansen, but still, and he was aware, Neisser was aware that Hansen was working on it, and even Hansen gave those samples which were infected with leprae bacillus. So he was quite upset with the with the action of Hans Neisser that he is taking undue credit for the discovery of the bacillus. Though still, but some again, what happened that Hansen attempted to prove his findings. Hansen attempted to transmit leprosy to a patient without informed consent later in his career was also considered unethical. So then in the later life of work on the leprosy, Hansen, without a mutual formal consent from the patient, which is a part of an ethical guidelines, he tried to inoculate the leprous bacillus, which is quite dangerous bacillus, and if the person is infected, may go through severe conditions or face some severe conditions in the body. But he tried to inoculate it. So this is one of the unethical action of the Hansen. Next important discovery was by Sir Alexander Ogston, year 1844 to 1929. a British surgeon. In 1881, Hoxton discovered Staphylococcus. Building on the teachings of his senior contemporaries, Louis Pasteur and Joseph Lister, Hoxton had observed pus from 88 human abscesses under his microscope and noted gram-positive spherical micrococci. Taking from the Greek word of bunches of grapes, he named the organism Staphylococcus, one of the most important pathogen which causes pyogenic infections in humans and skin, and mostly it infects the skin part. So the credit of discovering these organism goes to Sir Alexander Hoxton, and in the year 1881 he discovered the Staphylococcus. After injecting the isolated bacteria into healthy guinea pigs and mice and recreating the abscess from which the isolates were derived, he had conclusively introduced the world to the infectious agent now known as Staphylococcus aureus due to its golden color in culture that continues to burden human health today. Earlier, what happened that penicillin was the most active antibiotic against the Staphylococcus, but due to indiscriminate use of penicillin, the MRSA strains, that is the methicillin strain resistant Staphylococcus aureus strains, have emerged, and now it is becoming very difficult to treat those pathogens or eliminate them. Next important discovery was made by Frederick Loeffler, 1852 to 1915, a German bacteriologist, whereas Edwin Klebs, 1834 to 1913, a German physician and bacteriologist. So their findings basically was both were a German bacteriologist primarily known for their work on diphtheria bacillus, specifically their identification of the bacterium that causes the disease, also known as Klebs-Loeffler bacillus. Identified the diphtheria bacillus in 1883, Kleb was the first to observe the bacterium responsible for diphtheria in the pseudomembrane of infected patients. As a microbiology student, you would be aware that basically the diphtheria bacillus is responsible for the throat infections or the upper respiratory tract infections, and in the throat part they make a pseudo membrane, a covering along the throat part, which causes their which causes the choke of the trachea basically. So the first of the scientist which responsible for isolating from this pseudomembrane, which is one of the clinical manifestations of diphtheria. Clay was the first to isolate those bacteria from the pseudomembrane of an infected patients. Early inoculation experiment, he successfully inoculated syphilis in monkeys in 1878. Though he also worked on diphtheria, his work on syphilis is also greatly recognized. In 1878, he successfully inoculated syphilis in monkeys in 1878, predating later experiments, and produced tuberculosis experimentally in animals by injecting milk from infected cause. So he was not only his work is not only recognized for his for diphtheria bacillus, but he's also gets credit for his work on syphilis and tuberculosis. Then Klebsiella genus, as you know, that the genus Klebsiella, in the initial word Klebs is given there, and it has been given the name in his the Klebsiella bacteria name is in the honor of the scientist called Klebs. culturing the diphtheria bacillus. Building on Klepp's observations, Loeffler was the first to successfully isolate and culture the Corynebacterium diphtheriae bacterium in 1884. In earlier video, I have given you the about discussed about the Koch postulates, and in the Koch postulate, we have already discussed that there is four important postulate in the Koch. First was that the pathogen should cause a specific disease in an infected individual. Then it can be obtained in pure culture, and again, if inoculated in an experimental animal, it will cause same symptoms. And again, if it is isolated from the experimental animal, it will again be obtained in the pure culture. If we summarize in short, so when you will see these works, mostly they are inspired by the Koch postulates. So earlier they have, if you see the example of diphtheria bacillus, whereas the Klepp was the first to isolate it from the pseudomembrane of the infected patients. It is a characteristic symptom of diphtheria, whereas in the again it was obtained in a pure culture form based on the findings by Loeffler in the year 1884. Developed Loeffler serum. Next credit of Loeffler is that he created a coagulated blood serum medium, that is the Loeffler serum, also known as Loeffler serum slope, that is still used for the growth and differentiation of diphtheria bacillus in diagnostic laboratory. Next important scientist whose work is very highly recognized is Arthur Nicolaier, 1862 to 1942, a German physician and bacteriologist. In 1884, as a 22-year-old medical student, Nicolaier discovered the Clostridium tetani pathogen, the causative agent of tetanus, which is known in English as Nicolaier's bacillus and in French as Bacille de Nicolaier. Next important work is by Albert Frankel, 1848 to 1916, a German bacteriologist. For most of his life, he devoted himself to bacteriological research until he discovered the bacterium in 1886 that causes the pneumonia that bears his name, Frankel's pneumococcus. He excelled in the treatment of lung conditions, in which he became a true authority. In addition to a large number of work on pathology, physiology, and clinical observations, as well as an assiduous collaboration in the main scientific journals, he published diagnosis and common symptomatology of lungs disease. Basically, his work is mostly concentrated on the respiratory infections, and that also on the lung infections. And during his work, Frankel was able to isolate the pneumonia bacteria, which is also known by the name of Streptococcus pneumoniae, and in his honor, it is also named as Frankel pneumococcus. After that, next scientist is Sir David Bruce, year 1855 to 1931, a Scottish pathologist and microbiologist. Sir David Bruce's stories about his groundbreaking work on Malta fever, a disease that caused a major epidemic among British soldiers in Malta. In 1887, he discovered the bacterium Brucella melitensis in the spleens of deceased patients and showed it could be cultured. Later, Maltese physician Themistocles Zammit proved that the disease was transmitted through raw goat's milk, a discovery that was crucial for controlling the epidemic. The bacterium was later renamed Brucella in Bruce's honor. So the bacteria which is responsible for the Malta fever is Brucella melitensis, and he was the first to isolate it from the spleen of of an infected person, disease patient, and showed that it could be cultured. And then, based on his work, the Maltese physician Themistocles Zammit proved that the disease was transmitted through raw goat milk. Means that the source of infection is the raw goat's milk, and it was very important to control the spread of the bacterium. Next important contribution was by Fritz Sording, 1871 to 1906, a German zoologist, and Erich Hoffmann, 1868 to 1959, a German dermatologist. The primary contribution of Fritz Sordin and Erich Hoffmann was their discovery of the spirochete bacteria, bacterium Spirocheta pallida, later named Treponema pallidum, as the causative agent of syphilis in 1905. The story is such that on March 3, 1905, while working on at the Charité Hospital in Berlin, the German zoologist Schauding and the dermatologist Hoffmann identified the pale spiral-shaped bacterium Spirocheta pallida in fluid samples taken from a syphilitic papillae using a state-of-the-art microscope. Fritz Schauding, a geologist and a founder of experimental protozoology, made numerous other contributions to medicine before his early death at age 34. So main contribution is for in the credit of Fritz Schauding is the discovery of syphilis bacteria, that is Treponema pallidum, which is known today. But he has also other contribution, and mostly in the field of protozoology. He died at a very early age at 34. He was the first to distinguish between Intamiba histolytica, the cause of amoebic dysentery. Intamiba histolytica is a a protozoa, and the harmless Intamiba coli. So both are the two different species of Intamiba histolytica, whereas a pathogen means that it causes disease, whereas Intamiba coli is basically non-pathogenic protozoa. He helped confirm that hookworm infection occurs through the skin. Then in helminth, basically the hookworm infection, he proved that it causes infection through the by penetrating the skin. His studies on human and bird malaria parasite facilitated later work on the disease, confirming the findings of earlier researchers like Sir Ronald Ross gets the credit of in discovering the malarial parasite. But the earlier work was done on the birds and other animals, which inspired Sir Ronald Ross for his findings on malaria. Next is that about the Erich Hoffmann, 1868 to 1959, a dermatologist, continued his work in venereology and dermatology after the discovery. He became the head of the dermatology department at the University of Bonn, where he founded a large collection of wax models used for teaching medical students about various skin diseases. He was nominated for the Nobel Prize in Physiology or Medicine multiple times for his work related to syphilis. Next important scientist is Charles Jules Henry Nicole, year 1866 to 1936. He was awarded a Nobel Prize in Physiology or Medicine in 1928 for his work on typhus. Typhus fever is a disease that previously caused widespread loss of life during epidemic, especially in connection with wars and diseases. After it was established that several diseases, including malaria, were spread by insects, suspicions arose that this might also be the case with typhus fever. Charles Nicole noticed that sick people ceased to infect others when they had an opportunity to keep themselves clean. In 1909, he demonstrated that body lice spread typhus fever by successfully transferring the infection among apes by allowing a body louse to first bite infected and then uninfected apes. Next important discovery, recent discovery we can say, is by Barry J. Marshall, who is still between us, born in 1951, and Robin Warren, 1937 to 2024. Both are the Australian scientists. So Barry J. Marshall and J. Robin Warren's contributions were the discovery of the bacterium Helicobacter pylori and its role in causing gastritis and peptic ulcer diseases, a finding that revolutionized the treatment of these conditions. They demonstrated that ulcers could be permanently cured by using antibiotics to eliminate the bacteria, an achievement for which they were awarded the 2005 Nobel Prize in Physiology or Medicine. Today, Barry Marshall is the National Health Medical Research Council Senior Principal Research Fellow at the University of Western Australia. In recent years, his research has illuminated the patterns of Helicobacter infection in different populations around the world. It is probably the most widespread chronic infection in the world and is nearly universal in the world's poorest countries. Dr. Marshall hopes to see the insidious corkscrew organism controlled to the point where it can no longer pose a threat to the life and health of men, women, and children anywhere in the world. Dr. Marshall continues to practice as a gastroenterologist in Perth, Australia. See, one thing I would like to mention here that mostly the gastrointestinal infection is usually believed that it is caused by the lifestyle changes or through our eating habits. But Barry J. Marshall and J. Robin Warren uh, found the when he found that the infection is caused by a Helicobacter pylori, so it was considered that a lifestyle living a different lifestyle or unhealthy lifestyle is not only solely responsible for the gastrointestinal infection. So the bacterial in long time bacterial infection with Helicobacter pylori is also basically responsible, and it causes one of the most deadly gastrointestinal infection because it causes peptic, gastric, and peptic ulcers, which is quite dangerous. See, now next, I would like to, at the end of this video, I'd like to mention about two important Japanese scientists whose work is also very much commendable in the field of microbiology. That first scientist is Kitasato Shibasaburo, 1853 to 1931, a Japanese physician and bacteriologist. In 1885, Kitasato moved to Berlin to join the laboratory of German bacteriologist Robert Koch. There, with Emil von Behring, he studied tetanus and diphtheria, which we have already discussed about their findings by the scientists, two bacterial infections that cause symptoms through the secretion of toxins. So basically, tetanus and diphtheria themselves are not responsible, but because of their secretions called as toxins, which is also the poisonous substance called as named as poisonous substances. So Emil von Behring, with Emil von Behring, Kitasato came to the conclusion that it is basically through the secretion of toxins by these bacteria. In 1889, Kitasato succeeded in obtaining the first pure culture of the tetanus bacteria bacilli. See here also, we see that how Koch postulate greatly helped in the discovery of some of the very important bacteria. And the following year, he and von Behring demonstrated that immunity to tetanus could be achieved by injecting a susceptible animal with serum containing antitoxin produced in the blood of an animal exposed to the bacterial toxin. So it was also a major breakthrough that, as we see in the vaccines, that attenuated or heat-killed strains of bacteria give immunity in the individuals against that particular infection. Similarly, the toxins, if antitoxins are prepared from these toxins, they also helps in developing the immunity in the body of the individual and prevent those infections. And basically, they tested on those animals which were exposed to the bacterial toxins. They soon successfully applied this approach, and this approach was also known by the name of serum therapy to the treatment of diphtheria. Returning to Japan in 1892, Kitasato founded and became president of the Institute for Infectious Disease, a laboratory near Tokyo that was incorporated in 1899 into the Ministry of Home Affairs. The next year, he founded Yojoin, a sanatorium for victims of tuberculosis, and concurrently served as president of both organizations. Kitasato was sent to Hong Kong in 1894 to investigate an outbreak of the bubonic plague. Within a month, he identified the positive organism of the plague. See, usually the yersin, scientists here, rocks and yersin we have earlier discussed in the previous chapter. So, in the name of Yersin was the first scientist who discovered the plague bacillus, and in his memory or honor, the bacteria was earlier, which was called as Pasteurella pestis. Then again, it was named as Yersinia pestis, renamed after French bacteriologist Alexandre Yersin. But the work of Kitasato was also there on the plague bacillus, especially on the bubonic plague when he investigated an outbreak of bubonic plague in 1894. And he discovered the plague bacillus during the Hong Kong epidemic. Next important scientist whose work is Dr. Kiyoshi Shiga, 1871 to 1957, a Japanese scientist. Dr. Kiyoshi Shiga's main contribution include isolation of dysentery bacillus in 1897, which was later named Shigella dysentery, and coining the term Shiga toxin. He also developed the first vaccine for dysentery, including an oral vaccine that was administered to thousands in Japan. These achievements were foundational for the study of enteric disease and public health. So, basic work on which Shiga is credited is about the Shigella dysentery bacillus. And he also worked on the Shiga toxin. As already we have discussed, the toxins are the secretions of the bacteria, which greatly helped also in developing antitoxins. And based on that antitoxins, oral vaccines was administered to thousands of people in Japan. So Shiga was greatly credited for his work on Shigella dysentery, and also improving the case studies on enteric diseases and public health. So with this, I hope I have given some information on the work of the some of the important scientists whose work have greatly helped to improve the health conditions of the people, and to make a disease-free society. Thank you and good day.